Welcome everyone to the STEM student talk of the 6th Polygen Symposium of Rising Scholars. My name is Shreya Ravi Charla. I'm a second-year medical student at the Virginia Commonwealth University School of Medicine, and I'll be moderating today's talk. The goal of the symposium is to celebrate and showcase the hard work of all of our Polygen scholars, all of whom have tirelessly worked on their projects over the past couple of months. We have 4 student presenters presenting on a variety of topics today. A fellow Polygen's mentor will be scoring each student's presentation, and which will help us determine prize winners after the event. If you have any questions for the panelists, please make sure to type your questions in the chat, and we'll allot for 2 minutes after each presentation to answer those questions. So our 4 speakers today are Gabriele Barrocas, Nir Reiter, Simoni Mishra, and Siddharth Sivalanka. So with that, we'll get started with Gabriele. If you want to share your screen, and we can go ahead.
Okay. Okay, hi, my name is Gabriele Barrocas, and this is my presentation on my literature review titled The Age-Dependent Anatomical Abnormalities of the Amygdala in Autism Spectrum Disorder and Their Implications for Altered Socio-Emotional Development. This is just a table of contents talking about what I'm going to be talking about in my presentation, introduction, research, discussion, and a conclusion. Introduction. Autism spectrum disorder, or ASD, is a spectrum of conditions characterized by difficulties in social functioning, repetitive behavior, speech communication, etc. The range of symptoms that one individual with ASD has may not necessarily be the same symptoms another person with the disorder has. The amygdala is a bean-sized region of the limbic system in the brain and is responsible for processing emotions like fear and anger. When an individual is exposed to a fearful stimulus, information about that stimulus is sent to the amygdala, eventually triggering a response after the amygdala sends further signals. The purpose of my literature review was to examine the anatomical abnormalities of the amygdala seen in individuals with autism spectrum disorder that have a significant effect on symptoms and the development of the disorder. Volume is going to be my first subsection. Many studies have used structural and magnetic resonance imaging to examine the neuroanatomical abnormalities of the autistic amygdala. Research on this topic has shown that the amygdala is larger in young children with ASD compared to neurotypical controls, and that amygdala enlargement is often present by 2 years of age. One study specifically explained that the ASD group exhibited larger volume right and left amygdalae by about 14 and 12% respectively. It has also been reported that amygdala enlargement is positively correlated with the level of social interaction and communication deficits. However, using structural MRI scans, scientists have also claimed that as children with ASD grow older, their amygdala volumes end up being significantly lower than that of neurotypical controls. By adulthood, amygdala volumes of individuals with ASD are significantly reduced compared to controls, representing a reversal of the volumetric phenotype. The autistic amygdala appears to not undergo the age-related volume increase that is observed in typically developing children. The amygdala should increase in size from about age 5 to adulthood, about 40% in typically developing individuals, but low amygdala volumes are associated with the deficits of facial processing in ASD. Overall, after reading several studies, evidence suggests that increased amygdala volume compared to neurotypical controls is a characteristic of ASD in young children. ASD adolescents show no evidence of amygdala enlargement, as they do not go through the normal amygdala increase seen in neurotypical controls. More studies are needed to confirm this association. Additionally, numerous studies should research and elaborate more on the cause of abnormal amygdala morphology in individuals with ASD. My next section is on cell architecture and density in the autistic amygdala. Cell numbers in the autistic amygdala. Research on cell density of the amygdala as it relates to ASD started with Kemper and Bauman in 1993, who reported abnormally small and densely packed cells in the autistic amygdala. In 2006, Schumann and Amaral carried out an analysis of neuron numbers in the autistic postmortem amygdala concluding that there are about 11% more neurons in neurotypical controls in children, but about 20% fewer neurons in adults with ASD compared to controls. Additionally, in 2014, Morgan et al. Used post-mortem brains examining non-neuronal cell populations like microglia, oligodendrocytes, astrocytes, and endothelial cells. And this study found aberrant oligodendrocyte and microglial quantities in the amygdala of individuals with ASD. And if these specific cells are damaged, an action potential down an axon is reduced or seized. This figure is derived from Avino et al., and it basically just shows how neuron numbers decrease with age in 4 regions of the amygdala and in the total amygdala compared to controls. One study in particular, Wang et al. In 2018, used stereology to quantify the number of NNOS expressing interneurons in valproic acid mice models of ASD. NOSs are enzymes that aid in the production of nitric oxide, which is a molecule critical in cell signaling and neural development as it pertains to this case. NOS cells are GABAergic inhibitory interneurons, and evidence has suggested that deficits in these specific interneurons could be the pathophysiological mechanism of ASD. The scientists of this experiment concluded that the number of NNOS interneurons in the basolateral amygdala was reduced, also contributing to an excitation and inhibition imbalance, which plays a role in cognitive and learning deficits in ASD. And just a conclusion for this section, the autistic amygdala undergoes a postnatal development where the number of neurons reduces as an individual grows older. In the future, scientists should conduct more studies to understand the regulation of neural numbers that will allow for the interpretation of a normal role of the human amygdala during life. My last section is about connectivity in the autistic amygdala. This slide will talk about fMRI and how you can use that to study anatomical connectivity in the amygdala. The connectivity theory of ASD has attracted substantial curiosity in more recent years. It's been proposed that atypical white matter and connectivity patterns are indicative features of neuropathology of ASD. Multiple studies have used fMRI, functional magnetic resonance imaging, to examine the functional and structural connectivity between the amygdala and brain regions like the temporal lobe, striatum, cerebellum, which are all areas involved in social communication and also have to do with the repetitive behavior aspect of ASD. Amygdala occipital underconnectivity in ASD was observed, which could indicate inadequate integration of visual input into socioaffective circuits in the amygdala or in the brain as a whole. fMRI scans have also illuminated disruptions in the anterior commissure, a white matter structure responsible for linking the amygdala and the temporal lobes, but also contributing to memory, emotion, etc. Additionally, in individuals with ASD overall, altering the neuronal activity in the basolateral amygdala and social interaction. And for the last bullet point, additionally, young individuals with ASD have displayed weaker frontoamygdala functional connectivity, a difference that was more specific to the basolateral amygdala and its connectivity with the rostral anterior cingulate cortex, which is just an area that's implicated in socio-emotional processes and has an influence over amygdala-dependent learning. So overall, to sum up this slide, decreased connectivity in individuals with ASD leads to the decreased expression of the socio-emotional processes in an individual. This slide is going to be talking about more of DTI and interamygdalar connectivity. So DTI, or diffusion tensor imaging, allows for the analysis of fractional anisotropy, or mean diffusivity, providing outlines of white matter tracks in the brain. Mean diffusivity refers to the magnitude of water diffusion within brain tissue. So high values of this will indicate that there's increased water flow in the brain. Since DTI is an effective way to study neuroconnectivity, one study used this technique to examine the microstructural characteristics of interamygdalar connections and how they pertain to characteristics of autism. In this study, elevated mean diffusivity confirmed reduced densities of white matter tracks linking the right amygdala to the right cortex. Individuals with this elevated mean diffusivity actually had poor emotion recognition scores in certain tasks, to tie back to the whole autistic behaviors portion. And just a conclusion for this section, it is evident that overall connectivity between the amygdala and other regions, as well as within the amygdala, is disrupted in individuals with ASD. These weak connections are associated with increased autism severity and social functioning impairments. To sum up a little bit of this really long discussion, it is evident that overall connectivity between, this literature review is critical in understanding the anatomical basis of ASD, which is a developmental disability that affects about 1 in 54 children. Dr. Chun, the director of clinical research at Spark, stated in an interview, "Once you recognize the disorder in a child early, I think we have educators, behaviors, and other people who can help people to learn other ways to adapt. By understanding the amygdala, doctors can find strategies for intervention and improve the quality of life of the individual with ASD." After reviewing a series of anatomical and longitudinal studies, it can be concluded that the autistic amygdala suffers from multiple abnormalities that are not apparent in age-matched neurotypical controls, such as a reduction in neuron number contributing to increased anxiety levels, as well as weak connectivity associated with socio-emotional functioning impairments. These are my references. Thank you for listening.
Thank you, Gabrielle. With that in mind, does anyone have any questions for Gabrielle? You can either type them in the chat box or raise your hand. Okay, I have a question for you. Now that you have this foundation underneath your belt, what do you think you're going to do with the information you learned from this project and move forward with future projects you may have?
From the start of junior year, I've been focusing a lot on the advocacy of autism awareness, whether it be in school and in my community. So I also have a blog called The Real Spectrum, and I want to use all this information I learned to talk about more intervention strategies and simply like to uncover the why of autism. Why do people with autism act a certain way, and how can we create more of an inclusive environment for individuals on the spectrum?
Wonderful, thank you for that wonderful presentation. And with that, we'll go to our next presenter, Nir Reiter, if you want to share your screen.
Hi, can you hear me?
You're good to go.
Okay. Hello, I'm Nir Reiter. I'm a senior in high school applying for bioinformatics and engineering. Today, I will be presenting my research on chemical networks using a simulation I created, hoping to gain insights into the origin of life. So first, some background. The origin of life is one of the biggest unanswered questions in biology, with many competing hypotheses. One of these hypotheses relies on chemical networks, webs of reactions that interact through shared reactants and products. These networks are promising because they can form under chaotic starting conditions, but they have complex behaviors and react to changes in the environment. This property, complexity arising from chaos, is called emergence, and it is key in understanding how simpler chemicals became the intricate structures needed to maintain life. In order to study these networks and the origin of life, we will need tools to quantify and measure what life is. In particular, we want to measure how complex and stable the chemical network is. My goal is to develop these tools for measuring chemical networks and find a promising simulation to use them on. An early discovery in this field was the Belousov-Zhabotinsky reaction. The key aspect of this reaction is that cesium ions oscillate between two states, yellow and clear, creating spirals that radiate outward. The spiral structures form even under chaotic starting conditions and are incredibly stable, which makes this reaction an excellent reference for both emergence and for stability in lifelike structures. This is why I chose to simulate the BZ reaction. The reaction has been simulated before. Here is an example I took from Turner. Notice the spirals are visible, although they don't look exactly the same because the reaction has been simplified into only 3 chemicals and 3 equations shown at the bottom. An interesting simulation by Tsompanas used light to affect how the reaction behaved. On the left, you can see light shown in white and a mask which blocks light shown in black. The right image shows the simulated reaction, which still spreads in all directions, except under the mask where its behavior changes. The presence or absence of light created a change in behavior. Reacting to the environment is an important lifelike property, so I wondered, would adding light, but in a gradient instead of a mask, cause more lifelike properties to emerge? Energy gradients are common across different organisms, and they are more likely to arise naturally than a mask. With the presence of a variable energy source, more interesting properties may arise that may bring us closer to answering how life began. The simulation I created replicates Turner's method with the addition of an energy source. Like Turner, I used the cellular automaton, which is a grid of cells and a set of rules. Every step in the simulation, called the generation, the rules are applied to each cell based on its own state and the state of its neighbors to determine its state in the next generation. Most cellular automata have a small set of rules and possible states, which makes them easy to compute for computers. Despite this simplicity, complex patterns can arise from many interactions of cells. This makes cellular automata a useful tool for modeling emergence, complex patterns from simple rules. They do force the continuous real world into a discrete grid, but with a grid of sufficient size, this effect is minimal. I used a grid size of 100 by 100. I wrote my simulation in Python using a library called cellular automaton. My cellular automaton has 3 values ranging from 0 to 1 continuously, representing the quantity of each chemical in the cell. Here are the equations I used. For now, the coefficient L is kept at 1.0, so it has no effect. Every generation, chemicals are produced and consumed. Chemical A is produced from chemical B, B from chemical C, and C from A. This cycle maintains the oscillating pattern. Focus on the first equation. Net change in A equals amount produced from B minus the amount converted to C, all times the amount of A already present. The fact that A influences how much more A is produced is self-catalyzation. Every generation before applying these equations, I average the cell and its 8 neighbors to simulate diffusion, and afterwards I clamp the values to make sure they stay positive. To solve the problem of boundary conditions common in computer science simulations, I wrap the top side to the bottom and the left to the right. And here is an image of my simulation. This is the starting condition. I randomized it so that it starts chaotic. I chose to visualize all 3 chemicals, which I do by assigning each to red, green, or blue. The second image shows the simulation after 2000 generations. Notice how at first many different colors are visible, but eventually spirals emerge, and every cell is completely red, green, or blue. The cells clump together like this because of the self-catalyzing property of the reaction. Cells with lots of red will produce even more red, which will then be converted into blue and afterwards green, creating spiral structures that radiate outward. I needed to measure the reaction in a way that indicates how complex and stable it is. Out of the 4 different options I tested, I used the average entropy of regions 5 cells wide and 5 cells long. I call this measurement average local entropy. A value of 1.0 represents perfect chaos, a lot of change locally, while 0.0 represents perfect order, no change locally. Without looking at the simulation, we can see that the spiral structures formed after only 100 generations and afterwards remained very stable. Next, I added light, using a mask-like shape just as a test. Light is a coefficient, that's the L, which multiplies the rate of the reaction. The inner region has a coefficient of 1.0, while the outer region has a slower rate of 0.75. The outer region does not form spirals as quickly, which confirms that the light coefficient is working. Finally, I created a gradient by forming bands of light ranging from 1.2 to 0.4. Entropy is measured separately for each band. And now for the results. Here are some images. The first thing I noticed was that the gradient is visible even without me darkening the bands because the reaction flows along the gradient. So the gradient is definitely affecting the behavior of the reaction. At 500 generations, the spirals at the bottom seem to be the most prominent, but other local spirals are present in lower light bands. But after 3000 generations, the higher light spirals seem to take over. Notice that there is very little local behavior, very few spirals in the lower light bands at the top. This is kind of similar to natural selection. Spirals with more energy that can oscillate faster survive, while local spirals in upper bands fell victim to the waves. Here are the measurements from the simulation. On the left is a control group where I measure entropy for an entire grid at each light level to find a baseline. On the right is the entropy level of each band in the gradient. Notice that with more light present, the entropy decreases. This matches our biological expectations that with more energy present, more complex and organized structures can develop. The visual observations I made can also be seen in the graph. Focus on the 1.0 light band on the right. It starts very stable at around 0.77 in the control and in the experiment. But when exposed to a higher energy band, the entropy increases over time up to around 0.8. You can see that on the right. This appears to coincide with the later images, when the spirals have been replaced with flat weights. Just by looking at the graph, I could predict when the highest energy spirals began to take over. Average local entropy seems to be a good tool for measuring whether these spirals are present and for measuring the effect of energy gradients on the reaction. So from these results, I concluded that average local entropy is a reasonable measurement for stability, at least in the case of the simulation, because it matches the observed behavior of the reaction. This answers my first question: what tools can we use to measure these simulations? Second, I found that lifelike properties previously unseen in chemical networks can emerge in the presence of an energy gradient. In this case, survival of the fittest. This answers my second question. Would energy gradients have more lifelike properties arise? I also concluded that further research should focus on how early chemical networks used and benefit from energy sources. Maybe at the origin of life, energy was a key component in how life developed. Some ideas for future research: with more powerful computers, a larger grid could provide enough room for complex relationships. Expanding the simulation past this reaction could allow more complex behavior. You could model a different chemical network, a real one or a hypothetical one, and this is probably the easiest way to find more complexity. And finally, since the light band of 0.4 never developed any spirals, a good question to ask might be, what is the minimum energy needed for complex behavior? Thank you very much. Are there any questions?
Thank you, Nir, for that wonderful presentation. As always, you can throw questions into the chat or the specific Q&A feature, and we can get them to you to be answered. Alrighty, I have a quick question for you. I know you're talking about your further research. Have you thought about building on the current model that you have that you wrote in Python, or more of like starting from scratch to include all those extra variables?
Okay, well, it can build upon the model. If I go back for a second to these equations, you can see they're very simple equations overall. It's just three equations. They're all basically the same as each other. You can easily add just more of these, more chemicals if you want, that could add more different interactions, because currently only 3 different interactions are possible between the chemicals. But in real life, there's thousands, possibly even hundreds of thousands of chemicals that could be working together at the same time. So I think you can build upon this simulation very easily. I might recreate it to make it more efficient, so it's easier to calculate it. But yeah.
Wonderful, thank you. So for now, we'll switch to our next speaker, Simoni Mishra. So if you want to pull up your slides and we can get started.
Can you hear me?
Yep, we can hear you and we can see your slides.
All right, thank you so much. So my name is Simoni and I'm presenting a simulation study and diagnostic tool for early detection of Parkinson's disease. So what is Parkinson's disease? Parkinson's disease is, sorry, I got cut off for a second. Can you still see my slide?
If you want to reshare your screen, that might help with the slides.
Okay.
We can see them again, so we're good to go.
All right, thank you so much. Sorry about that. So what is Parkinson's disease? Parkinson's is a disorder of the central nervous system that affects movement. So some of the early symptoms of it include hand tremors or slow movement or facial masking, something that I will be focusing on. And facial masking or hypomimia is basically the reduction of a person's ability to move their facial muscles. So broadening out again, more than 10 million people worldwide and 1 million people in the United States live with Parkinson's disease, although there's no definitive test that exists to diagnose the disease. This means that if a patient comes to their physician saying with X number of symptoms, the physician is likely to just extrapolate from their medical history, signs and symptoms, etc., to decree, I guess, that they have Parkinson's disease. But this is slightly counterproductive in the fact that by the time the symptoms have shown up, you're already pretty late in the progression of the disease, which means that a lot of the pharmacological and non-pharmacological treatments out there are way less effective than they would have been if you would address them earlier on. So that's what my project is basically tackling, looking at ways that we can, it's actually taking advantage of facial masking in order to diagnose Parkinson's disease as early as possible. So taking a look at facial masking or hypomimia, it's a condition in the earlier stages of the progression of Parkinson's disease that limits the movement of facial muscles, restricting an accurate depiction of facial expressions. It's basically facial muscle movement rigidity. And then taking a step to the side really quickly is an important concept to my presentation, something called the facial action coding system, or FACS. And basically what this is it's a comprehensive system describing all facially discernible movement through a fundamental unit called the action unit. And what an action unit is, it's basically a set of different facial muscles that together do something. So for example, they could raise your they could part your lips or raise your cheek, et cetera, et cetera. And these are some of the examples. And so the idea is that if you combine various action units together, you get what what we call expressions like smiling or disgust, et cetera. So moving back for a second, we're just going to look into my procedure. And so for this project, I've basically had 3 main steps. 1, I'm collecting data. 2, I'm creating my model. And 3, I'm testing my model. So how did I create my data? So when I first approached this project, I came in looking for data sets of the facial images of Parkinson's patients. However, I wasn't able to find such things online, so I decided to simulate that. And I did this with a software called OpenFax. It's an open source FACS-based software that allows for the simulation of facial expressions. And like I explained earlier, what this does is it has an array input, essentially, and the items in the array are all corresponding to various action units. And as the user, you input the intensity of those action units, effectively creating some sort of facial muscle. And I generated all of these different facial expressions, essentially for both Parkinson's patients and non-Parkinson's patients, by looking into earlier research papers and grabbing the action units that they had recorded for these specific groups. And so looking into the data set itself, at the end of the day, I generated about 22,000 different images, both Parkinson's cases, with 11,000 for Parkinson's cases and 11,000 for control cases. And I covered about 4 different facial expressions, including disgust, surprise, neutral, and happy. Of all of that data, 75% was split for my training data set, and then 25 was kept for my test data set. And that will come up a little later. One thing about generating my data, I was simulating my data myself, and I realized that using this software, OpenFax, I was only able to generate the faces, but it was much more difficult to save them because the engineer of the software engineer, hadn't really put in a method for me to like download or like save the images. So I got in touch with them. But at the end of the day, what I did was I used OS commands, os.import to really just screenshot the page and save each of them. And I did this in an algorithm because of course, 22,000 images takes a while. Okay, so going into model creation. So I created a CNN model, or a convolutional neural network, which has specialized applications in image recognition. And so when I first created my model, it turns out it just didn't run. It required a much more powerful processor than what I had. And so it took about 2 days to give me any sort of output. So the first thing I had to do off the bat was talk to some people, and I ended up changing my computer for a computer with a GPU, or a graphics processing unit. And even doing so, it did improve the processing speed, but it was still quite slow. So what I then did was I realized that the output images that OpenFacts had given me had a large boundary width. So like, sure, they had the facial expression, but they also had a large width that was just like nonsensical information that my model did not need. So what I did was I resized all of the images into a 150 by 150 pixel square. And that also, among other things, that helped increase the processing speed. So at this point, I have a model that runs and I'm like, yay, but then I realized that it's giving very arbitrary outputs. Regardless of whether I input a Parkinson's or a control image, it just kind of goes on a 50-50 and gives me whatever it thinks it is. So accordingly, I had to change my model, and in specifics, my model architecture. So I learned a lot about the layers used within my model architecture, and I layered them in specific ways. I ordered the layers, which you can see are the convolutional layer, the pooling layer, and the fully connected layer. And I also messed with their parameters, messing with the batch sizes to see how that affects the efficiency and the accuracy of the model. And I did this looking at the accuracy and efficiency with two other tools, namely TensorBoard and Matplotlib, using their graphing features to really get a sense of what is going on in the model. I also added a activation ReLU thing because my images were nonlinear. So this effectively helped really helped gauge, just improve the accuracy of my model. And after doing all of this, I got to a point where my training set worked really well. And so I was very confident and moved on to my testing set and realized that the accuracy from there just plummeted dramatically. And unfortunately, this was due to something called overfitting. My model had gotten way too comfortable with my training data set and was not yet ready to move on to a testing data set. So originally what I had was about 100 epochs. My model is running at 100 epochs, which means that it was iterating about 100 times. So it was getting way too familiar with my training data set. So I decreased that down to 50 epochs. And I also added additional dropout layers, which are said to help with overfitting. And doing this, and along with a few other things, eventually helped me get my accuracy up to 94%. After messing around with this, I also went and looked at the VIT model, or the Vision Transformer model. This is known right now to be, it's like a new and upcoming model used for originally text classification, but now also image classification. And so I began to use this, but quickly realized that my data set was not as large as this model requires. And so while the other one had like a accuracy of 94%, this model only gave me like up to 80%. So conclusion, the whole point of this study is really to make diagnosis of Parkinson's disease as accessible as possible. And so that people don't have to wait until it hits the last stage where the symptoms are overwhelmingly powerful that they approach their doctor and get diagnosed and receive treatment. So my goal is to take this model, and I've already started to do this, but create a mobile application for it, and utilize the camera features on a mobile device in order to automatically diagnose Parkinson's disease. So yeah, and that, along with the simulating images, is quite useful in a general context outside of Parkinson's disease, as when you're not using human subjects, you're saving a lot of time and money. So thank you so much for listening to my presentation. If you have any questions, I would love to hear them.
Wonderful. As always, you can enter your questions in the chat box, use the raise hand function, or the Q&A function, and we can get those questions answered. I have a question, as always. So I know you mentioned that you were trying to get this into a mobile app form that can be used in clinics to easily diagnose those early sets of Parkinson's disease. How do you envision any struggles translating from the mostly AI generated set to implementing this in real life?
So I've already kind of implemented this in real life, where currently the stage in the mobile application is such that I can take an image in a pre-existing camera roll and put that into my model, which is on the device, and that will result in some sort of declaration of Parkinson's or not. So that is working. I do imagine there being more difficulties in the future as I test this more. This has just kind of been a side passion of mine to kind of go into it and see what happens. So it hasn't been fully tested, but yeah, I totally do anticipate there being differences between the AI generated data set in comparison to real people. But I think that just would change as you get more data, maybe by collaborating with clinics or things like that.
Wonderful. Thank you for your presentation. And with that, we'll move on to our final speaker for this STEM section. Siddharth Sivalanka, if you want to share your screen and we can get started.
Yeah. Is my screen visible?
Yes, it is.
And my voice is good? Or
Yep, your video and your audio are great as well.
Okay, I'll start. Good morning, everyone. My name is Siddharth Sivalanka, and my polygenic mentor is Miss Ashley. My research focused on genetic disorders, their formations, and their treatments. My polygenic project was to research these disorders, and I formed a website to display this information. Before moving on to the genetic disorders that I researched, though, I want to start by recapping some basic information that could be helpful to know. So genetic disorders. Genetic disorders are caused by changes in the DNA, usually through mutations. There are various types of these, like there's point mutations, chromosomal mutations, deletions, insertions, and hence they can be good, bad, and sometimes even neutral. And an autosome is any chromosome that is not a sex chromosome. So there's 23 pairs of total chromosomes, and chromosomes 1 through 22 are called autosomes, and chromosome 3 is a sex-linked chromosome. And alleles are one or two forms of a gene on the same chromosome. So on chromosome number 4, the same kind of gene from one from mom, one from dad, that'd be an allele. And these can be dominant and recessive. So dominant means that it's expressed more, and recessive means that it would be masked or hidden away. And so to take this into context, for example, in a fly, if we have green eyes as our dominant trait and pink eyes as a recessive trait, unless both the parents carry the gene for the recessive allele or the pink eyes, there's always going to be green eyes. And one more important thing I want to add on is that a polygenic disorder is a disorder where multiple genes have a part in the disorder, whereas monogenic means just a single gene, only relying on one. So now, since we covered the sort of basics that would be helpful to understand, at a quick glance, this is what I'll be talking about. So first I'll be talking about Huntington's, then I'll be talking about sickle cell, and then finally asthma and an effect of a very emerging and good treatment. So we're going to start off with Huntington's. Huntington's disease is a type of genetic disorder in which the central nervous system dies actually, and it causes the behavioral and psychiatric issues. So as we can see here, as demonstrated in the picture of the brain, we can see that the area affected right here is called the basal ganglia, which is responsible for your body to have smooth motions and control over what you're doing. Huntington's is essentially a single gene disorder involving the HTT gene on chromosome number 4, as said right here. The main job of this protein is to send signals to the brain, which is why it's commonly found in nerve cells, because that's the job of the nerve cells. So as we can see in the image on the right, Huntington's, it's a very, very delicate mutation. As we can see, it's just this red part highlighted. And what happens is this is the gene and it has an increased amount of glutamine repeats. So glutamine is a CAG codon. And so if it's more than 35, you go to Huntington's. And what happens is, this is a polypeptide chain. So it's like the unfolded version of a protein essentially. And so as you can see, the polypeptide chain is extended and it's very long. And what happens is the neuron is not formed correctly, and then the nerve cells don't work well, and the function of the entire thing collapses. And so, as mentioned earlier, this revolves around the brain, meaning that the symptoms that it causes are relying on the motor features. So, for example, like walking, talking, swallowing, reading, these kind of symptoms. Other symptoms include mood swings, poor memory, loss of interest, and even depression. Sadly, though, even with the advanced 21st century technologies that we have, we still can't solve Huntington's because of its complexity. And so the only treatment we have for now is to ease the life of the people, but due to in future research, scientists are trying to find ways to solve this. Next, we'll be moving on to sickle cell anemia. So sickle cell anemia is a genetic disorder, which is only a single gene as well, so on the HBB gene, and follows an autosomal recessive pattern. So before we move on, I want to talk a little bit about hemoglobin. So what hemoglobin is and the details of it. So hemoglobin is made essentially out of 4 chains, 2 alpha, as said right here, and 2 beta chains right here. And so if any of these chains are disrupted, the protein will not fold properly, and it won't be able to do its job, which is to be able to transfer oxygen throughout the body. So if we look at the image over here, we can see that, so this is the tRNA bringing the amino acids. And so what happens is this is the genetic material here, and this is what happens if there's sickle cell. And the only difference here is the A changes to a U here. And so basically what happens is the glutamine here changes to another amino acid called valine. And what happens is, even though this is a very small mutation, the whole polypeptide chain and all the folding of proteins changes. And due to this tiny error, instead of turning out like a donut shape, it eventually turns out like a moon kind of shape. And so what happens is, again, hemoglobin can't function properly, and that's what causes sickle cell anemia. And so sickle cell has various different symptoms, but they can include swelling, infections, delayed growth, vision problems, and most deadly of all, jaundice. And so previously, there were really no treatment to sickle cell, but due to 21st century technology, we finally found a solution. It's called gene therapy, which will be discussed in a later slide. And it's an issue that's fixing this treatment. It's been very effective. Finally, we're going to move on to asthma. So asthma is essentially polygenic, so it's multiple genes, and it has more than 100 genes at play. So it's a very complex disorder that scientists just cannot find what to do with it. But they're all centered around chromosomes 5, 6, 11, 12, and 14. So these are all autosomes. So as described on the picture over here, we can see that this is chromosome number 5. As I said, it's mostly on 5, 6, 11, 12, and 14. The only changes we have here is the AT bond, right? AT nucleotide bond here is changed to a CG. And so the most important gene is the ORMDL3 gene. It's essentially, not in the complexities, essentially what it does is it makes you have trouble breathing. And as we can see with the airway inflammation here, this is a normal airway with someone who doesn't have asthma. And so in a normal airway, we can breathe properly and the air circulates well. But with someone with asthma, demonstrated by the red here, it's a little more condensed, but they still have the air can still circulate. But during an attack, you can see that the muscle that says here is tightened, which signifies the fact that it's tight and you can't breathe properly and the circulation is not good. And so asthma is actually influenced by various environmental factors like allergies, ozone gas, air pollution, etc. Symptoms of asthma include shortness of breath, coughing, trouble sleeping, and asthma's treatments are mostly lifestyle changes like taking dietary measures or reducing exposure to like flu. So as said before, I'm going to talk about gene therapy. So gene therapy is a very effective treatment that's been used for a lot of genetic disorders. Throughout my research, I looked at various different treatments like the CRISPR treatment with the cutting of the vagina and replacing it. But the most appealing to me was gene therapy, and it's a very viable treatment. And so as you can see here, they used a vector, usually a virus, to put it into the human body, which starts making the good proteins and it fixes the genetic disorder. Well, that's a really oversimplification of it. So if we look at a more detailed picture, stem cells are taken out of the human, and then we use viruses and they're removed of the genetic material, and you put the genetic material that you want. And viruses need host cells to reproduce. So they attach to a host cell, they put their genetic material in, and when you inject it, more proteins that you want are formed. Thank you so much for listening to my presentation. Any questions?
Thank you, Siddharth. As always, we'll take questions in the chat box or via the Q&A, or if you want to use the raise hand function, we can take the questions that way. All right. I had a quick question. Out of the 3 different diseases you discussed, Huntington, sickle cell, and asthma, do you have any indication of which would be easier to treat with this new gene therapy?
Yeah, so gene therapy treatment, as mentioned earlier, it works for sickle cell anemia. So the issue we're running with Huntington's is the problem that scientists are trying to use the same idea to try to kind of solve it, but due to the complexity of the disease on naturally, it's not working. But luckily for sickle cell, it does work because sickle cell, the problem is the hemoglobin doesn't form properly, and when we introduce these proteins that help form hemoglobin, it does form properly, and therefore it can fix sickle cell anemia and start producing good hemoglobin. So your oxygen can flow through your body and keep your body healthy.
Cool, wonderful. Thank you for your presentation. And with that, we'll wrap up this STEM talk. Thank you for all the attendees who came and listened to our wonderful Polygen students and the hard work that they put in. And we hope that you enjoy the rest of our symposium today. Thank you.